Thank you, everyone, for joining us today for Mapping Molecular Landscapes, how EMSL's spatial metabolomics capabilities can accelerate your research. Today's webinar will focus on EMSL's innovative approach to analyzing metabolites spatially and the EMSL capabilities that you can request for this approach. Our next EMSL LEARN webinar is going to be held on March 11th and will focus on decoding complexity through EMSL's single-cell multi-omics capabilities, such as nano and microsplits, as well as nanopots. You don't want to miss this webinar. So more information will be provided on our website and through our social media channels, as well as via email. Make sure that you're keeping in touch with us by subscribing to our email and our social media channels if you haven't already. And links are being provided in the chat. So for today, Dušan Veličković is an EMSL chemist who will focus on spatial metabolomics of the rhizosphere and microbes using the MALDI-MSI, revealing metabolic interactions in native environments. We have with us, joining us today is Fiona Ellsworth, a PhD candidate at the University at Buffalo, and she will discuss how she's used EMSL's RhizoMAP capability, a non-destructive platform for metabolic imaging, to advance her recent project. Then you'll hear from EMSL chemist Marija Veličković, who will highlight the laser capture microdissection-metabolomics, which is a technique detailing technique for isolating and analyzing specific cell types or regions. And we have Che Clendinen, who also is an EMSL chemist, and she will walk us through the nano liquid chromatography-MS setup and data analysis workflows that are essential for processing spatially resolved samples. There will be time at the end of today's webinar for questions, so we encourage you to post your questions in the Zoom chat so that our speakers can answer them during the Q&A. And with that, we will begin with Dušan.
Thanks, Linda, and hello, everyone. Today, I will talk about our capability that we offer to EMSL users, and basically using MALDI mass spectrometry imaging for spatial metabolomics. But in this case, I will focus how we use this MALDI mass spectrometry imaging capabilities for analyzing microbes and rhizosphere. So today, what you will learn actually, and it will be really good introduction for all other speakers that you will hear today, is some basics of mass spectrometry imaging, and then some also basics and principle of MALDI mass spectrometry imaging, and then how we employ those MALDI mass spectrometry imaging techniques for spatial metabolomics at EMSL, and I hope you will find use of those techniques, how you can implement them in your research in future calls. So I will focus on imaging, for example, of microbial interactions when we grow microbes on the agar plates in the petri dish. Then how we use MALDI mass spectrometry imaging to image rhizosphere metabolites within synthetic soil habitats that we call terraforms as well. And then how we actually image extracellular metabolites within native soil environment, like using a RhizoMAP approach. And we will have Fiona after me that will really provide you more detailed information how you can employ this RhizoMAP approach in your research. So I will not spend much time here. I will let her talk about it. So basically, when we talk about mass spectrometry imaging, we talk about spatial probing of sample surface chemistry by mass spectrometer. So if you see here on the figure, where we have tripartite community composed from different microbes and also moss as well, we can go from position to position in the sample, probe every position of the sample by different probes. I will go later there. And then from each of the position of the sample, we can acquire mass spectrum, and that mass spectrum actually reflects molecular composition in that particular area that we sampled. And when we finish our mass spec imaging analysis, basically when we probe each position in our sample, we end up with those ion images that actually reflects relative abundance of specific molecules across the sample surface, where each, in this case, color corresponds to different ion in the mass spectrum, and that ion corresponds to specific endogenous molecule in the sample. So as such, this allows us to reveal spatial distribution of molecules within a biological sample. We can design our experiments to be completely untargeted, so we don't need to a priori know what molecules are present in the sample. But if you, for example, want to focus on some specific molecules, we can even, it's even better for us, we can target our analysis for specific class of the molecules and enhance their sensitivity. And you can see here that by multi-mass spectrometry imaging, we can really analyze big array of molecules, so all the way from small molecules via metabolites, lipids, sugars, glycans, even small proteins. Today, I will mostly focus on the metabolomics, as this is topic of this workshop. And then, depending how well we can focus or what probe we use in our mass spec imaging approach, we can have different spatial resolutions, so which can go from let's say cell clusters, and you will hear today in Maria and Che talk how we can use relatively large spatial for relatively large spatial information on the probe in the techniques called, for example, liquid extraction surface analysis. But we can also focus our probe to a single cell level, to let's say micrometer or 5 micrometer scale, and get information from the single cells. And really, EMSL is equipped with numerous tools that can enable those imaging analysis. And today, I will focus mostly on the MALDI mass spectrometry imaging capability, because it is definitely most employed tool for molecular analysis, for analysis of native molecules in the sample. And this MALDI stands for matrix assist laser desorption ionization, where essence is that we add molecules that called the MALDI matrices that kind of enhance ionization of your molecules and help in ionization of your molecules, and also absorb energy of the UV laser that we use as a probe to spatially probe the sample surface chemistry. And in MALDI, basically, we can offer and reveal different type of metabolites. So let's say from amino acids, molecules of TCA, pathway, phytohormones, phenolics, alkaloids, sugars, lipids, biogenic amines, etc. And as I said, requisition here is that we need to coat a sample with a MALDI matrix, and that MALDI matrix, as I said here, will kind of move or decide what molecules of those that I listed here we will visually. So it is important that we know if we will do targeted or untargeted analysis, because those matrices show selectivity towards specific analysis. And in MALDI, we usually operate at 5 micron or 20 micron spatial scale, although we can, if samples are very large, we actually analyze them with, let's say, 200 micron lateral resolution. So it depends from the sample, from the size of the sample for scientific question. But in routine analysis, we do let's say 20 micron, but we can go down to 5 micron. We use a couple instruments here at EMSL. One is called FTICR, or Fourier transform ion cyclotron resonance instrument, that provides us ultra high mass accuracy, so we can really be confident in the molecules or in molecular formulas that we see. And then we have also MALDI timsTOF that has a high spatial resolution and high throughput analysis. So it's roughly like 20 times faster than FTICR. However, it has like limited mass resolving power or mass accuracy. So we usually in our approaches use both of those instruments to address scientific question. So in microbial interactions, we use often molecule or often samples or microbial colonies that are grown on the agar plates, and then usually like different interaction between like fungi, bacteria you guys are interested in. And what we can do, actually, we can dissect piece of the agar where this interaction occurs. Doesn't need to be interaction, can be single colony as well. And then we put this piece of the agar on our specific target and basically profile it with the MALDI-MSI. Often we use approach that involved on-target chemical derivatization, where we, besides using MALDI matrix that kind of helps in the absorption of molecules, we can increase ionizability of the molecules by, let's say, modify them using, by covalently modify them. In this case, for example, if you use this is like approach that developed in our lab using specific compound called 4AP, but that can modify carboxylic acids or derivatized carboxylic acids and aldehydes, put permanent charge on them, and help them to be actually, and make them actually always positive ions. And in that sense, we can really cover roughly like almost 400 lot of microbial and secondary metabolites in plant as well are carboxylic acids or aldehyde group, and we can cover roughly like 400 metabolites by this approach. Here I show you like one figure from our paper where we can really just a small, in this case, just a few examples of the molecules that we can detect in different areas of interaction between Bacillus subtilis and Fusarium, but there is many more, as I said, like 400 metabolites, and then you can make kind of conclusions of how those microbes interact in a specific environment and scientific question you guys want to answer. So then we can also go and analyze, let's say, molecules to the depth of the biofilm, for example. So we can take, as you can see here, biofilm of the Bacillus subtilis. We can cross-section it, mount it on the glass slide, and then basically profile those sagittal sections of the biofilm and get information how phenotypes are changed inside the biofilm. And you can see here from the either from the surface to the bottom of the biofilm or from the periphery to the center, and we can again detect a lot of different molecules. Some of them also exuded in the agar. One of them is, for example, also like Bacillibacterin, important siderophores. But also what we can do is even like go and analyzing signaling peptides by this approach and analyzing a little bit bigger molecules than just metabolites. So another approach what I want to highlight here is how we can use mass spec imaging for analyzing rhizosphere metabolites. And this is for, I don't know if you guys had the opportunity to listen some of webinars about terraforms, but one of terraform that we extensively use in mass spec imaging is actually rhizochip, which are those synthetic soil environments that kind of mimic soil pores or channels between soil particles, and they're basically made from the materials that are compatible with mass spec imaging. So we have like PDMS where those pores are made, and then we kind of overlay this rhizochip with a specific membrane that is actually very suitable for mass spec imaging analysis. And here we can have like control conditions. Either we will inoculate with some bacteria exposed to different food sources, stresses, etc. And this is that workflow what we use for rhizochip analysis and analysis of those exudates of the rhizosphere, where we basically put the seedlings in those PDMS for design device, let it grow, but remember that here we actually on top of it have that specific membrane that we, after growth of the plant is done, we kind of peel that membrane off, and that membrane has basically imprinted molecules from those pore channels from the root surface, and then we put that membrane basically on the specific again MALDI target plate, and either use like on target chemical derivatization or not, depending what we want to analyze, and basically analyze it by MALDI FTICR or MALDI team stuff. You can see here that Megan put some of the links for the Terraform's capabilities if you want to look it more there. So this is some of the examples that we can see there. And for example, you can see how we can go and analyze and actually visualize distribution of really different molecules that associate either with the roots or different root types, or they are just exclusively present in the specific pores of the or channels between the soil particles. We can even go and zoom there and see like really some hot spots that actually are highlight microbial activity. There, in this case of Burkhold area, that was inoculated in vicinity of those hot spots, so we can kind of get information about metabolism that is occurring in the microbes, but also what root exudates and how microbes kind of change that root exudation in depending of what conditions we set. And I want to finish this part with RhizoMAP approach that we also develop at P&L and offer to EMSL users, and you will hear from Fiona on the next talk, how we can use, how we can actually image root exudates microbial activity as well within native soil environment. Remember, rhizochips were synthetic soil environment. Here we have like native soil environment where we grow the plants in the rhizoboxes, and then again use different imprinting techniques to transfer molecules from the rhizosphere to the membrane, and then mount that membrane on the again specific MALDI target plate, which will then either again do, for example, chemical derivatization or spraying specific MALDI matrix, and basically do spatially profile then with using MALDI FTICR capability. As a result, we have like images that looks like this, for example, where we can profile molecules to the molecular depth or to the spatial depth, for example, from the root base all the way to the root tip. And then we can also see how different molecules, for example, associate really closely to the root, but others are kind of diffuse into the soil. And again, here we can detect basically, again, roughly like 300 to 350 different primary and secondary metabolites, mostly that has like carboxylic group on it. What is also very cool here is, and you will hear that later as well, is that this is also allows you to track exudation and processes inside the rhizosphere in time, because this is undestructive technique, and you can place those membranes and remove them in different point of time if you want to monitor, for example, development of the root growth or whatever process is occurring in the rhizosphere. And with that, I would like to thank you, and also I would like to introduce Fiona, who will kick off from here.
Thank you, Dušan. My name is Fiona Ellsworth. I'm a PhD candidate at the University at Buffalo. And as Dušan said, today I'm going to be talking about an application of RhizoMAP in our lab that we've done via an EMSL user project to investigate root exudation under environmental change. So this might be a review for some of you, but the rhizosphere is this area of the soil that's impacted by plant roots. And in this figure from Kuzikov and Razavi, we can see the myriad ways that roots impact their soil environment, including by changing gas concentrations, soil porosity, moisture levels, and biological activity. And one of the really important drivers of this impact that roots have on their soil environment are root exudates, these carbon-containing, chemically diverse biomolecules that plants transport from their roots to their surrounding soil. And so understanding root exudates is really important for understanding broader rhizosphere biogeochemical cycling and even broader soil biogeochemical cycling. So we specifically wanted to know how conditions of hydraulic redistribution impact root exudation from corn plants, Zea maize plants. So we normally think about water leaving plant roots via their leaves in the process of transpiration, but during the nighttime when transpiration is shut down, we can have this movement of water from wet to dry areas of the soil via plant roots. So that's being shown here in this figure from Amanu and Kumar, this process of hydraulic redistribution in the light blue dotted lines in this figure, where during the nighttime, water is moved from these more moist, deeper soil layers to drier surface soil layers via these plant roots. And we wanted to know how this process of hydraulic redistribution of HR impacts corn root exudation. So under non-HR conditions, exudates leaving plant roots comes up against this opposing flux of water entering plant roots. But under HR conditions, we hypothesized that water leaving plant roots would carry more exudates and carry these exudates further into the rhizosphere. And so to test this hypothesis, we grew corn in rhizoboxes shown here in this image. And then used this RhizoMAP root imprinting technique that Dušan introduced to you all to collect exudates. So here I'm showing you that membrane placed up against the root. We would incubate this membrane there for a time so that when we take the membrane off, it now has exudates collected on that membrane with their spatial distribution preserved, and we can analyze them via mass spectrometry imaging techniques. Specifically, we used MALDI-FTICR MSI. And what was, I should note, what was interesting about this project was that we trialed this novel membrane for this root imprinting technique, anodic aluminum oxide membranes, which enabled analysis of both nonpolar and polar metabolites, and then also enabled collection of exudates across this whole range of soil moisture conditions due to the very fine pore structure of anodic aluminum oxide membranes. So we could collect exudates under both droughted and well-watered conditions. So I'm now going to show you all the results of this experiment. Here we're looking at the same set of corn roots, but on the left, these are those roots under non-HR conditions, and on the right, these are the roots under HR conditions. And now I've overlain our chemical imaging results, specifically from malate onto our root images. So these yellow pixels are corresponding with higher malate signal intensity in our chemical images. And I've added white boxes over the same set of root tips under non-HR and HR conditions. And we see under non-HR conditions a pretty thin, narrow footprint of root exudation of malate. But under HR conditions, there's this much expanded exudation footprint of malate, this much wider footprint of malate exudation under HR. Now I'm showing you hydroxybutanoic acid. Very little was imaged under non-HR conditions, but we have this much expanded exudation footprint under HR. This is citrate. We had some imaged under non-HR conditions, but somewhat more, a somewhat larger exudation footprint under HR conditions. And then finally, now I'm showing you oxoproline. So we actually had a larger oxoproline exudation footprint under non-HR conditions compared to HR conditions. And oxoproline is this plant drought stress response metabolite. So this makes sense that we would see expanded footprint of oxoproline under non-HR versus HR. So I've shown you all just 4 of our chemical images that we collected, but we imaged a couple hundred of metabolites for each of our samples. So our next step was to really reduce the dimensionality of this data and to pull out some top-line conclusions, some top-line stories about how exudation was impacted by hydraulic redistribution. So to do that, I performed a principal component analysis on all of our chemical imaging data. And I'm here showing you the results of that first principal component, which captured most of the variation in our data, nearly 70%. And so each column in this figure, A through E, corresponds to a root sample. And the top row are those roots under non-HR conditions, and then the bottom row are those same roots under HR conditions. I have subset these chemical images to just the rhizosphere, so just a couple millimeters out from the root. And I've annotated where the roots were on our membranes with these darker gray lines. And then I've also drawn boxes around our root tips to draw your eye towards these differences in exudation that we see under non-HR and HR conditions. And so we're looking at the scores of the first principal component here. So these brighter blue pixels correspond with our exudation signal. And from our principal component analysis, we found that HR drove this expanded exudation footprint for many of our roots. So for example, looking at our root sample E here, there's somewhat of an exudation signal under non-HR conditions, but this footprint is much expanded under HR conditions for those same set of roots. And so now I'm showing you root samples F through J. And so for 7 of the 10 roots that we sampled, we saw this expanded exudation footprint under HR conditions compared to non-HR conditions, as indicated by our principal component analysis scores. Another result from our principal component analysis are the loadings that drive our first principal component. So these are the metabolites that are most responsible for the patterns that our principal component scores show. So here we're looking at some of the strongest loadings from our principal component, 6 metabolites here, and we found that these are all primary metabolites, simple primary metabolites, organic acids, amino acids, simple carbohydrates. These are the metabolites that are responsible for that expanded exudation footprint under HR conditions. So in conclusion, we trialed this novel anodic aluminum oxide membrane with RhizoMAP to measure both polar and nonpolar metabolites under a whole range of soil moisture conditions, and then used this method to answer this question of how hydraulic redistribution impacts exudation from plant roots, and found that it drives this expansion of exudation at root tips, and that primary metabolites, these simple metabolites, are the ones responsible for this expansion of exudation. And this suggests that hydraulic redistribution may be this hot moment of exudation for at least corn in our study. And I'd just like to acknowledge all my collaborators and thank you all.
Thank you for joining us today. My name is Marija Veličković, and I am EMSL chemist, and I've been with PNL 5 years and mainly working on spatial multi-omics sample preps. And today I'm gonna show you some capabilities that we use for profiling metabolites with a spatial context. And although Dušan and Fiona already explain you some basics of MALDI-MSI spectrometry imaging, and they show you some very interesting results of exometabolites, I'm going to start my talk with, again, MALDI-MSI spectrometry imaging, because I want to also show you the pipeline that we have for plants, in case you're interested in endometabolites and in case you want to know what is going on intracellularly. So in case you want to study plants, either you are interested in the roots, stem, or leaf, we can also image them, and the first step for us would be to do embedding of your sample. So we would freeze and embed the sample in order to basically support the structure of the sample, and that's really important for really rigid and delicate and highly water content samples such as plants. And for embedding purposes, we use hydrogels such as hydroxypropyl methyl cellulose. That's the hydrogel that is commonly used for our pipeline, and that's basically help us with cutting the tissue and prevent tearing, and also preserve really nicely morphology of the plant. So first step would be to embed, freeze, and then cryosection. And once we cryosection our sample, we would mount the cryosections on the slides that are used for MALDI imaging, and then we would image our section and obtain an ion image, such as have like nice ion image of the root here. And here in EMSL we have ultra high mass analyzers that can allow us to really obtain accurate molecular mass of our metabolites, and we can also, based on that molecular mass, ascribe molecular formula, and since MALDI imaging is MS1 based technique, under this particular molecular formula, sometimes it can be only one molecule, but in this particular case, under this molecular formula, you can see that there are 7 molecular candidates that can be there. So that's why we very often use some orthogonal technique that can provide us MS2 compositional level, and that can help us to really confirm the metabolite ID. And from the orthogonal techniques that we very often use here in EMSL, we use bulk metabolomics that can provide us MS2 identification, or we use some other spatial metabolomics techniques. So for bulk metabolomics, basically, we would need to take entire root to freeze it, grind it, and extract the metabolites from there. And then those metabolites we can run on the mass spec and then obtain all the metabolites that are present into our sample. And some disadvantages of our bulk metabolomics is that we really require a lot of starting material, especially if we're comparing with the MALDI imaging pipeline where we need just a tiny piece of sample. And also another thing with bulk metabolomics is, yes, it's great, we have those metabolites ID, but we lose the spatial information with bulk metabolomics, and we kind of really want to understand what is going on at the spatial level. So that's why some of the techniques that we use for orthogonal MS2 confirmation are LISA MSMS and LCM SMS techniques. And in my talk today, I'm gonna show you really in detail those two techniques. So the first one is liquid extraction surface analysis mass spectrometry capabilities, or as we call it, LISA. And from now on, I'm going to refer to this technique as LISA. And LISA is an ambient extraction techniques that can extract the metabolites from the solid surface in the automated fashion. So first step would be basically, if we have a cryosection, to place the cryosection on the slide. And then after that, the robotic arm would withdraw our extraction solvent, and then it would go in the certain position with a certain X and Y coordinate from where we want to extract the metabolites, and basically in that analytical spot we would come with a robotic pipette and basically dispense the liquid. Once we dispense the liquid, we would make really liquid microjunction that would hold for a few seconds. And then after that, once we extracted the metabolite, we would withdraw the extracted metabolites and then directly infuse in our mass spectrometer to obtain MS1 and MS2 data and basically to profile our sample. And LISA is the capability that can it can be used like standalone technique, but so we can basically use the LISA to image entire tissue section like I have it here, or it can be used in a targeted fashion when if we have maybe MALDI imaging data and we are interested in confirming IDs, we can maybe focus in some specific regions that we only want to sample, and we can look for the certain metabolites if we know which metabolites we are interested in. And I just want to now show you a few examples of using LISA. And first example is basically how we coupled MALDI and LISA. In fact, it's better to say how we use LISA to validate MALDI imaging result for exploring interkingdom interaction within the microbiomes. And for this particular experiment, we had a three-parted system where we had plant, fungus, and bacteria that were grown on the agar. And we excised those three-party system, like dissected that three-parted system and move it to the ITO slide. Then we also place the control from each bacteria, fungus, and bacteria, as well as agar blank control, and we dry that sample. After that, we perform high spatial resolution multi-mass spectrometry imaging to really to spatially resolve metabolites and to look at the metabolites with a really high resolution. What is, as I said, for MALDI imaging, we can really have a great spatial resolution going down to a 5 micron, but we are still getting only MS1 and we are getting only tentative annotations. So that's why we use in this case LISA approach as the orthogonal technique, where we sampled the same slide with limited spatial resolution, which was in this case 150 micrometer, but we obtained MS2 data from each of those samples, and then we were able to confirm the IDs for the metabolites that we imaged with really high spatial resolution by multi-mass spectrometry imaging. Another application of LISA, and this is a little bit different, is that we can couple our LISAs to our SubTAP capability. SubTAP is the capability that we also have here at PNNL, and SubTAP stands for substrate tapping, and that's the basically capability that we have for rapid analysis of exometabolites that are resulted from plant microbe or microbe interactions. And as you can see here in this image, SubTAP basically is a 3D printed platform, and it contains 3 pieces. The first piece is basically growing chamber that we can customize and make it however we want it to suitable the best our application. Then the second piece would be polycarbonate filter, and the third piece would be a 96 well plate that would collect all the excreted metabolites. And as you can see here in my example here, we use SubTAP basically to investigate microbe interaction when we had in that first piece that is growing chamber, we place it 2 microbes, and then we were able to track the exometabolites, so the metabolites that were excreted down there by collecting in that 96 well plate. And we also in this example we were looking at the plant microbe interaction, and we were collecting underneath the excreted metabolites. And what I basically forgot to explain you, that for those 96 well plate, when we collect those excreted metabolites, we dry them, and then each of those well, basically we extract the metabolites and then inject them into the extract them by LISA technique, and then inject them in our mass spec to obtain MS1 and MS2. And that's how, with the spatial context, we can investigate plant microbe and microbe interaction using subtab coupled to LISA system. And now I'm gonna talk about that other capability that we also have for metabolomics profiling with the spatial context, and that's the LCM metabolomics capability. LCM is a capability by itself, I have to say, and LCM is a sampling technique that we use for isolation of a single cell or population of the cells. And here, as you can see in my slide, if we start from sorghum crop and we use one little stem, embed it in cryosectioning, this is how the cryosection looks under the microscope. And we can, under the direct visualization, under the microscope, we can basically look at the subpopulations of the tissue cells. And as I have it here under the 20x objective, we are looking for in the sorghum step, we are looking at the vascular bundles and also cortex. And with the laser capture microdissection, we can excise and collect the specific cell types that we are interested in. And here I outlined that we were interested in the vascular bundle. So basically we would first outline the regions we are interested in, and then with the laser capture microdissection, the UV laser, really focused laser, narrow laser, would excise that region, and then with the next pulse of the laser, we would catapult that excised region and collect into collection device. And for collection device, I have here a tube cap, but we can also use some nanopod chip and micropod chip, because this is a capability, LCM capability, that we also use for spatial proteomics and spatial transcriptomics. And if you want to find out more about that capability, please join our next seminar in March. But for our LCM metabolomics capability, this is how the workflow looks like. So the first step is to do cryosectioning and obtain the cryosection. After that, once we know which specific cell types or regions we are interested in, we can excise them and collect them into the tube cap. After that, in the tube cap, we would add the extraction solvent and then turn it upside down, like keep it like that upside down, and then sonicate it for a little bit to extract the metabolites. And the next step would be to centrifuge that, to spin down the liquid and place our tube into the LCM vial, and then we would inject our extracted metabolites on our mass spec system. And I'm gonna now turn it over to Che, and she's gonna show you some really cool results that we were able to obtain using our LCM metabolomics capability.
Hello, everyone. Thank you for that, Maria. My name is Che Clendinen. I'm a chemist here at EMSL, and I'm just going to be going over a couple results from our LCM-based nano-LC metabolomics capability, which is relatively new. So one of the results that we've had is we collected some stem cortex, epidermis, and vascular bundles from, and Maria showed these to you on the previous slides. And we were able to detect about over 500 features, as well as most of them having MS2, and identify about 68 of those features. And then using this, we were able to show the PPCA as well as correlation maps, that there are clear differences in the metabolic profiles across these different tissues. And some of the metabolites that we were able to see do come from primary metabolism, including amino acids and sugars. So this is just one example of what we can now do when we have that LC separation ahead of the MS2 detection. We also are working with some of researchers like yourselves. This is a relatively new project, and so the data is still project is still ongoing, but we have been able to capture over 2,000 MS features with more than 100 putative identifications, as well as MS2. So we can see things like, again, the amino acids. And there's particularly, Jamie Jamie Bowers-Rios at the University of Missouri, is particularly tracking lignin biosynthesis and distinct cell types of the sorghum stem. So using the method that Maria had described, we can actually collect very rich data from a small amount of sample, as you can see clearly here on the bottom. Again, this is still ongoing, but it's really exciting that we're actually starting to work with some of you in the audience. But there are some things that you need to keep in mind. So there are some practical considerations. A sample type. This is really work well for plants or microbes. If you do have other sample types that you are interested in, please connect with us to see if it can be compatible with this method. The metabolites that it detects are quite diverse, but we do use a C18 column. So you're likely going to get a really good better separation of those semi and non-polar metabolites, so we do detect those more polar metabolites like amino acids and sugars. We run it in 2 modes, positive and negative, and can definitely give accurate identification of metabolites, since we do have retention time in MS2. But if there are metabolites that you are particularly interested, it would require you to provide standards, or we sometimes can have standards here at the lab that we can run to accommodate that. And the quantification is relative quantification, so it's not absolute. So we are comparing across samples the different those particular metabolites. And so that's just a really quick and fast example of what we are able to do with this relatively new technology. And then we're really excited if any of you guys in the audience, please contact us if you are interested in seeing if your research is compatible with anything that was presented. These are the point of contacts here at the lab that have been the forefront of all of this research.
Fantastic, everyone. Thank you so much for your presentations. And at this point, we are going to move on to the Q&A portion of this webinar. All right, so the first question is directed to Maria. It looks like Trevor Lewis is asking, under MS1 annotation, up to 7 candidate molecules can be considered. With MS2 annotations, can isomers be differentiated? Often isomers with the exact same M over Z have different biological functions. How are isomer relationships considered, or are they combined with parent features? So it's a two-parter.
Well, yeah, I think it's a great question. Yes, we can distinguish the isomers, but I think I would basically address this question to Che, because she's our data analyst, and yeah.
So the benefit of having the liquid chromatography prior to the mass spec is that we can have some separation of isomers and isobaric compounds. Now, there are plenty of isomers, and the example of a good example would be sugars, like hexose sugars. They give almost the exact same MS2 features. So some isomers are going to be very difficult to distinguish and would require a bit more upfront isolation prior to putting it on our platform, just because we are using a C18 column and not all isomers can be separated chromatographically with our current method. And so while the answer to your question is yes and no, depending on the isomers and their chemical properties.
All right, cool. Trevor has another question that is directed towards Fiona. So you had mentioned that several primary metabolites were the cause of the root exudation expansion. Could these metabolites just be the byproduct of the hydrated environment? Is there other reasoning to support this besides the large variation in PCA? Fascinating research, by the way.
Yeah, thanks for that great question. So when I say cause, yeah, I just mean that our principal component analysis ascribed that pattern of expanded exudation footprint under HR to mostly being driven by those primary metabolites. But that, yeah, that's only one one line of evidence. We also collected bulk carbon data from these same routes under HR and non-HR conditions. And so we have total carbon exuded, and then we also have, I'm currently running LC-MS on those total carbon samples. So I don't have the LC-MS data for you on those bulk carbon samples. Our total carbon shows a similar story of HR driving this increased exudation. So somewhat corroborating our PCA analysis. So I think I'm getting at your question. The LC-MS, which we are running right now, might be able to further clarify what are these metabolites that are driving this expanded rhizosphere.
Awesome. Thank you, Fiona. All right. So we have Gayatri Sharma is asking Dušan, this is for you. How can MALDI-MS be used to spatially resolve low abundance quorum sensing molecules at microbial interfaces? And what are the main challenges?
Oh yeah, good question. So I think, main challenge in MALDI actually is that, like we don't have any chromatographic separation here or enrichment that we can do, right? So we just shoot with the laser and everything that is there will go in the mass spectrum. So we can do like some kind of derivative. We were never like really focused on quartz. I know that there are like some lactone stuff that we were like kind of trying to do it. It's very hard to preserve lactone structure, by the way. So we tried like different derivatization approaches. So for example, for some of the peptides molecule, it's not so challenging, but main challenge is we don't have any chromatographic separation. We cannot do it like in selective way, and the main challenge is they are very low abundance. So we are probably like somewhere in femtomole abundance that can detect, and it need to be relatively ionizable molecules. So we never really like had any projects that were like kind of going into specific quorum sensing molecules, but those are challenges that I foresee. But yeah, happy to talk more detail about that later or whenever.
Okay. So this one isn't isn't for anyone specifically. So jump in if you know if you have an answer. So Ram Singh is asking how to store the agarose plugs pieces for analysis. Suppose for bacteria interactions, for chemo attractants and repellents. So
I can jump in because it's probably like related to what I show on one of the slides. So what we usually do is, for example, users they set up like bacteria interaction or microbial interaction on agar. We send them our slides, so it's and then they dissect it and place it on the which we call on that specific glass slide that we send them, and then they ship them back to us as a in vacuum seal bag, like completely dried and vacuum sealed, and then we go from there. Because we tried before, like that users send us entirely like agar plates with microbes and stuff like that. It didn't work really well in that direction. So what we usually recommend is that we send you the slides where you will actually kind of mount those pieces of the agar plugs, how you call it here, and then after we have like a SOP for that, dry them down, vacuum seal, and send it in our lab for analysis. It's pretty easy actually to manipulate with those agar plugs and place them on the glass slide.
Excellent. Okay. We have 5 more minutes left for this webinar. So again, if anybody in the audience has a burning question for our folks here, please drop them in the chat. The next question that came in, so for you, Maria, does the subtap approach require samples to be sent to EMSL, or can experiments be performed in our lab?
Yes, great question. So we can definitely 3D print it and send those subtabs to the users, or we can do the experiment here in EMSL. So either user can come and do, or we can do the experiment. We kind of here in EMSL, we have a growth chamber where we have built-in humidifier chamber. So really, we don't need to water the plant if we're growing some plant, and also in that chamber we can maintain humidity and sterility. So yeah, that's what we can do here. And as I said, a user can come and help us with experiment, or if they want to like do an experiment on their own, we can 3D print those plates, subtabs platform and send them.
All right. So Abigail is wondering, Dušan, how can MALDI be used to study rock in its powdered form? How can it be used to study rock microbe interactions? I am interested in studying the metabolite that the microbes use in leaching metals from the rock powder.
Oh yeah, this is actually a great question, especially like, so if rock is in the powder, then I don't see any issues there, because like we can, so in MALDI, I forgot to mention that, I think Maria kind of hinted that we need to have like kind of flattish surface there, so we cannot do really like if sample has topography, but if we have powder of the rock, we can just place the powder on like our specific target that has like copper tape. We can glue it on that copper tape that is conductive, and I don't see any. I mean, we analyze like also like kind of soil particles in that way. So I also analyze like a few other like not rock but powder compounds. Just put. I mean, you kind of lose spatial information obviously, but you can analyze it from the solid material directly. So it's actually pretty straightforward in terms of sample prep. Now, like what molecules we will detect, that I mean it depends on specific problem, but in terms of workflow, it's not like it's relatively trivial. So
Great. Awesome. Okay, so again, another one that anyone can jump in. Ram is also asking, so regarding RhizoMAP, do we need to perform the experiment at EMSL, or can we perform it in the real field and do the analysis ourselves? How to do the sampling and the process? Does anybody have
I mean, I can say like from for example, or Fiona also can talk there, but Fiona's experiment, I mean, okay, some of Fiona's experiments that showed here were performed in the EMSL rhizo boxes, but some were performed actually in her lab, right there. So what like you need to send to us are only those membranes, right? So and it is because we have capability, like we have like MALDI FTICR and that instrument that is really like dedicated to that. But you can do it in your own lab, like Fiona, like most of her data she showed today were performed in her lab. What you need to send to us are only those imprints. So we also like kind of design something to be performed in the field as well. But keep in mind, like we also, what Fiona showed also, like you want to have like optical image as well that you can overlay with your rhizosphere and stuff like that. So how you will do it, you can make like rhizobox in the field as well. We saw those like installation as well, but you can do it. It doesn't need to be performed in EMSL. So I don't know, Fiona, if you have something else to add there, but
No, exactly. Yeah, we just we had to preserve the membranes right after we collected them in our lab here, just via like vacuum desiccation and then freezing. But then we shipped them and they were still analyzable.
Very cool. Okay, Dušan, Abigail has a follow-up question about the powdered rock.
yeah.
She was wondering, is there any way you can obtain spatial information from the powdered rock?
I mean, we will get information. So first one, like remember, like our scale of analysis is like 5 micrometers, right? So we cannot go like and see. So whatever powder sample, however we put the powder on the on our tape, we will get spatial information of those particles that are there. But spatial resolution there will be like 5 microns. So let's say like you put like 3,000 particles of that powder there, we will have like information we will have like spatial information which will be like 5 micron spatial information. So not anything that you can do, for example, with nanosims and stuff like that you can do in nanometer and things like that. So in terms of that, yes, you will have like spatial information on that bulk powder that you put there, right? So but is it like in the native, is like powder particles, are they like native in the same orientation like they were in your native sample? It depends just how you will place them on the target plate. I don't know if that kind of answered your question, but you will get spatial information. It will be like 5-10 micron spatial information, and image will depend on the way how we put that powder on the target multi-target plate.
Okay. We did have one more follow-up question from Trevor, but we are over time on the webinar, so we understand if attendees need to drop off. Trevor is wondering, for plant leaf cryosections, what's the best embedding matrix? That he's used CMC, but at low concentration, between 2 to 4%, as to not rehydrate the plant material. So is anybody able to very quickly?
I can take that question. So yeah, we use HPMC, 7.5% HPMC with 2.5% PVP. That's hydroxypropyl methyl cellulose with polyvinyl pyrrolidine, 7.5 HPMC and 2.5 PVP in water. And we used also for embedding and cryosectioning of the leaves, and it gave us beautiful cryosections.
Fantastic. All right, and with that, we end another EMSL LEARN webinar. I want to thank everyone in the audience for joining us today. We appreciate you coming, attending, asking these fantastic questions. I see Ram is asking, when can we see the video? In a few weeks, or in about 2 weeks or so, we'll have the video up on our website for you to review. So yeah, have a great day.